Chapter 7. Downstream chaos. Downstream chaos emerges when early-stage constraints are carried forward into the small intestine, where precision is required but no longer possible. The intestine depends on receiving material that has been structurally modified, chemically conditioned, and delivered in controlled amounts. When those conditions are not met, the intestine does not fail in isolation. It attempts to compensate for upstream deficits using mechanisms that were not designed to handle poorly prepared substrates. The result is a system that remains active but loses coordination, where breakdown, absorption, motility, and microbial interaction begin to diverge from one another. The first point of disruption appears in enzymatic breakdown. Pancreatic enzymes are calibrated to act on substrates that have already undergone partial reduction in the stomach. When larger, less processed proteins and fats arrive, enzyme efficiency decreases because access to cleavage sites is limited. Enzymes are present, but the structure of the material prevents consistent interaction. Some regions are reduced effectively while others persist, creating a mixture of fragments that vary in size and chemical composition. This variability is the starting point for downstream instability. Bile coordination becomes less effective under these conditions. Lipids that have not been properly exposed or dispersed require more extensive emulsification, yet bile delivery depends on signaling that assumes a certain level of preparation. When this assumption is not met, bile may be insufficient relative to the state of the substrate. Fats remain in larger aggregates that resist enzymatic action, reducing the formation of transportable units. This alters not only fat absorption but also the physical environment within the intestine, influencing how other nutrients are processed. Motility adjusts in response to these altered substrates. The intestine uses feedback from the lumen to regulate transit, attempting to balance exposure time with processing capacity. When breakdown is incomplete, the system may slow movement to increase contact time or accelerate transit if irritation or osmotic effects are detected. These adjustments are reactive and often inconsistent. The timing between secretion, breakdown, and absorption loses alignment, and the sequence becomes less predictable from one segment of the intestine to the next. Absorption efficiency declines as a direct consequence of these upstream and local disruptions. Nutrients that have not been reduced to their absorbable forms remain in the lumen, passing through transport regions without being taken up. The intestinal lining is presented with substrates that exceed its capacity to extract, leading to increased loss of usable material. At the same time, larger fragments and partially processed compounds continue moving distally, increasing the load on regions that are not optimized for primary digestion. Microbial interaction intensifies as unprocessed material reaches the lower intestine. Substrates that would normally be absorbed earlier become available for fermentation, altering the composition and activity of microbial populations. This changes the chemical environment within the gut, producing compounds that can influence motility, barrier function, and immune signaling. These effects are not isolated to the microbial layer. They feed back into the host system, further increasing variability in digestion and absorption. The system attempts to stabilize itself through compensation, but these compensations often introduce additional variability. Increased enzyme secretion, altered motility patterns, and shifts in fluid movement can partially offset the initial deficit, yet they do not restore alignment. Each adjustment is made in response to conditions that originated upstream, and the underlying sequence remains disrupted. The intestine continues to function, but it operates in a state where coordination between its components is reduced. Downstream chaos is therefore the cumulative expression of early misalignment. It is not caused by a single failure within the intestine, but by the propagation of constraints that began before the stomach completed its work. When early phase signaling, acid timing, and gastric coordination are restored, the substrate entering the intestine changes. It arrives in a form that matches enzymatic and absorptive capacity, allowing the system to operate with consistency. Without that restoration, the intestine remains in a reactive state, continually adjusting to inputs that do not align with its design. Sequential failure propagation. Digestion operates as a dependent chain where each stage inherits the output of the previous one, and any deficit introduced early is carried forward as a structural constraint rather than a transient error. When the initial phases begin below capacity, the system does not reset at the next stage. It builds on what it has been given. Weak sensory activation lowers early secretory output, delayed acid limits protein unfolding, and incomplete gastric processing delivers substrates that are not aligned with intestinal requirements. Each step receives material that is already compromised, and the sequence progresses with that constraint embedded in the system. The propagation begins with altered substrate structure. Proteins that have not been fully unfolded retain regions that resist enzymatic access, and fats that have not been properly exposed remain in aggregates that limit emulsification. These structural features are not corrected automatically downstream. Pancreatic enzymes act on what is available, but their efficiency is governed by access to specific sites. When access is restricted, breakdown becomes uneven. Some components are reduced while others persist, creating a heterogeneous mixture that continues through the intestine with varying levels of preparation. This heterogeneity drives secondary adjustments in secretion and motility. The intestine attempts to match its output to the state of the material it receives, increasing enzyme release where possible and altering transit speed to extend or reduce exposure time. These adjustments are local responses to upstream conditions. They do not restore the original sequence, but attempt to compensate within a system that is already misaligned. The result is a pattern where different segments of the intestine operate under different conditions, each responding to the same substrate in its own way. As partially processed material moves further along, the burden shifts to regions that are not designed for primary digestion. Larger fragments and unabsorbed nutrients reach distal sections of the gut, where microbial populations interact with them. This introduces additional variables into the system, including changes in luminal composition and chemical signaling. These changes feed back into motility and secretion, further altering the behavior of the digestive tract. The sequence becomes increasingly complex, with multiple layers of response acting on a substrate that was never properly prepared. The key feature of this propagation is that each stage amplifies the effects of the previous one. A small deficit in early signaling can lead to a larger disruption in gastric processing, which then produces a more significant mismatch in the intestine. The system does not fail at a single point. It accumulates deviations from alignment, and those deviations interact with one another. By the time material reaches the distal gut, the original cause is no longer visible as a single event, but its effects are embedded in the behavior of the entire sequence. Reversing this pattern requires intervention at the level where the sequence begins. Strengthening early signaling, restoring acid timing, and aligning gastric coordination change the structure of the substrate before it enters the intestine. When the initial output is corrected, each downstream stage receives material that matches its design parameters, and the need for compensation decreases. The sequence regains continuity because each step is now building on a properly prepared input rather than attempting to correct an inherited deficit. Compensation across the intestine. When the intestine receives material that has not been properly prepared upstream, it does not stop functioning. It shifts its behavior in an attempt to manage the load, adjusting secretion, motility, and absorption in ways that partially offset the deficit. These compensations are driven by local sensing of luminal contents and are applied segment by segment. The intestine is not correcting the original problem. It is redistributing effort across its length to maintain forward movement and extract what it can from a substrate that exceeds its design processing conditions. Enzymatic compensation is the first response. Pancreatic output can increase in response to the presence of larger or more complex substrates, raising the concentration of digestive enzymes in the lumen. This increase improves breakdown where access is available, but it does not change the underlying structure of resistant regions. Enzymes act where they can reach, leaving portions of the substrate only partially reduced. The result is a mixture that still contains fragments too large or too complex for efficient absorption, even though enzyme activity has been elevated. Motility adjusts in parallel. The intestine modulates transit speed based on the chemical and physical characteristics of the contents it detects. When breakdown is incomplete, transit may slow to extend exposure time, allowing enzymes more opportunity to act and absorption mechanisms more time to engage. In other situations, particularly when unprocessed material alters osmotic balance or irritates the lining, transit may accelerate. These opposing adjustments reflect an attempt to stabilize the system under variable conditions. The sequence becomes less predictable because transit is no longer governed by a consistent processing timeline. Fluid movement is also altered as part of this compensation. The intestine regulates secretion and reabsorption of water to maintain a workable environment for enzymatic activity and nutrient transport. When substrates remain in larger or more complex forms, fluid dynamics can shift to facilitate dispersion or to manage osmotic gradients. These changes influence the consistency of the luminal contents and affect how enzymes and bile interact with the substrate. Variability in fluid handling contributes to fluctuations in how efficiently nutrients are extracted. Absorptive mechanisms operate under these altered conditions with reduced efficiency. Transport systems in the intestinal lining are optimized for specific molecular forms. When substrates have not been fully reduced, these transporters are underutilized because the required forms are not present in sufficient quantities. The intestine may increase surface interaction through changes in motility and contact time, but the limitation remains structural. Larger fragments continue moving forward, carrying potential energy that is not being captured. As these compensations occur, the distribution of workload across the intestine shifts. Proximal regions attempt to handle a greater burden, while distal regions receive material that would normally have been absorbed earlier. This redistribution introduces new interactions, particularly with microbial populations that encounter substrates in forms and quantities that differ from baseline conditions. The chemical environment of the lumen changes, and these changes feed back into motility, secretion, and barrier function. Compensation across the intestine, therefore, reflects a system attempting to maintain function without restoring alignment. Each adjustment addresses an immediate constraint but introduces variability that affects subsequent stages. The intestine continues to process and absorb, but it does so under conditions that are less stable and less efficient. The underlying sequence remains disrupted because the initial preparation of the substrate has not been corrected, and every compensatory action is layered onto that original constraint. Motility disruption and unpredictability. Intestinal motility is organized to move material forward while maintaining alignment between secretion, breakdown, and absorption. This coordination depends on receiving substrates that have been processed to a predictable state. When upstream transformation is incomplete, the signals that govern motility become inconsistent because they are derived from a substrate that varies in structure and composition. The intestine adjusts its movement based on what it detects in the lumen, and when that input is unstable, the resulting motility patterns lose coherence. Segmental contractions and propulsive waves are normally timed to allow enzymes to act and nutrients to be absorbed before material advances. When partially processed substrates enter the intestine, this timing is altered. Larger fragments and unevenly reduced components trigger local responses that either slow movement to increase exposure or accelerate it when the system detects conditions that require rapid clearance. These opposing adjustments can occur within the same region over short intervals, producing a pattern that shifts between retention and rapid transit. The sequence no longer follows a consistent rhythm. Chemical signaling contributes to this variability. The presence of unprocessed proteins, lipids, and other compounds influences the release of mediators that regulate smooth muscle activity. When substrates are not in their expected forms, these signals do not align with the standard progression of digestion. Motility is driven by inputs that fluctuate in both intensity and timing, leading to contractions that are not synchronized with enzymatic activity or absorptive capacity. The intestine continues to move contents forward, but the relationship between movement and processing becomes unstable. Fluid dynamics further interact with these changes. Variations in osmotic load and luminal composition influence how water is secreted or absorbed, altering the consistency of the contents. Changes in consistency affect how easily material can be propelled and how it interacts with the intestinal wall. Thinner contents may move more rapidly, while thicker or more heterogeneous mixtures may be retained longer. These physical differences feed back into motility patterns, adding another layer of variability to an already unstable system. The cumulative effect is unpredictability in transit time and processing efficiency. Material may pass through sections of the intestine too quickly for complete enzymatic breakdown and absorption, or it may remain longer in other sections without achieving the necessary transformation. The system oscillates between states rather than maintaining a stable progression. Each adjustment is a response to local conditions, but those conditions are shaped by upstream deficits that have not been resolved. Motility disruption therefore reflects a loss of coordinated timing across the digestive sequence. Is not a failure of movement itself, but a breakdown in how movement is regulated relative to processing and absorption. When upstream alignment is restored, the signals that govern motility become more consistent, and the intestine returns to a pattern where propulsion and processing are synchronized. Without that alignment, motility remains reactive, and the sequence continues to operate with variability that cannot be stabilized at the level of movement alone. Microbial shifts and fermentation. The composition and activity of microbial populations in the intestine are determined by the substrates that reach them. When upstream digestion is aligned, most proteins, fats, and simple carbohydrates are reduced and absorbed before they can serve as fuel for microbial metabolism in distal regions. When early processing is incomplete, larger fragments and unabsorbed nutrients move forward, altering the substrate field that microbes encounter. This shift changes which populations expand, how they metabolize available material, and what byproducts are produced within the lumen. Fermentation increases when substrates that should have been absorbed earlier remain available. Microbes break down these compounds into smaller molecules, generating gases and organic acids that alter the chemical environment. The rate and pattern of fermentation depend on the type of substrate present. Partially digested proteins, residual carbohydrates, and unprocessed fats each support different metabolic pathways within the microbial community. As these pathways become more active, the balance between microbial groups shifts toward those that can utilize the available material most efficiently. These changes influence the physical state of the intestine. Gas production affects luminal pressure and can alter motility patterns, while organic acids modify pH and influence the activity of both microbes and host enzymes. The intestinal lining is exposed to compounds that vary depending on the substrates being fermented, and these compounds can affect barrier function and signaling pathways that regulate immune and neural responses. The system responds to these changes, but the responses are shaped by the altered environment rather than by the original design of the digestive sequence. The timing of substrate delivery also affects microbial behavior. When gastric emptying and intestinal motility are inconsistent, substrates arrive in irregular pulses rather than in a steady flow. Microbial populations respond to these fluctuations, expanding when substrate is abundant and contracting when it is limited. This variability contributes to instability in the microbial ecosystem, where shifts in population dynamics can occur over relatively short periods. The chemical outputs of these populations then fluctuate in parallel, influencing motility, secretion, and absorption. Microbial shifts, therefore, represent a downstream expression of upstream inefficiency. The intestine is not only processing nutrients for the host, but also managing a secondary system that responds to what remains unabsorbed. When digestion is aligned, microbial activity is supported by a controlled and limited substrate supply that maintains stability. When digestion is disrupted, excess and inconsistent substrates drive changes in microbial composition and metabolic output, introducing additional variability into the system. These changes feed back into the host, affecting how the intestine regulates movement, fluid balance, and barrier integrity. The system adapts to the altered microbial environment, but these adaptations are layered onto the original disruption. The result is a digestive sequence that is influenced not only by incomplete upstream processing, but also by the secondary effects of microbial metabolism acting on substrates that should not have reached that stage in their original form. Immune activation from incomplete digestion. The intestinal immune system is positioned to respond to material that reaches the epithelial surface, and its level of activation depends on how completely that material has been reduced before arrival. When digestion proceeds with proper sequencing, proteins are broken down into small peptides and amino acids that can be transported across the epithelial layer with minimal interaction with immune surveillance systems. When upstream processing is incomplete, larger fragments persist within the lumen and come into contact with the mucosal interface. These fragments present structural features that are more likely to be recognized as signals requiring attention, increasing the likelihood of immune activation. The barrier between the lumen and the internal environment is selective, relying on both physical structure and transport mechanisms to regulate what passes through. When substrates are in their intended forms, they are absorbed through controlled pathways that limit exposure to immune cells. Larger, partially digested fragments do not follow these pathways efficiently. They may remain in the lumen, interacting with the epithelial surface, or they may cross in ways that engage immune detection systems. This interaction is not random. It is driven by the structural properties of the material that has not been fully reduced. Local immune responses are triggered when these interactions reach a threshold. Cells within the intestinal lining detect patterns associated with incomplete digestion and respond by releasing signaling molecules that influence both immune activity and epithelial behavior. This can alter barrier function, change fluid dynamics, and affect motility. The response is designed to manage potential threats, but when the stimulus is persistent, the system remains in a state of ongoing activation. This shifts the baseline condition of the intestine, affecting how it processes subsequent meals. The presence of partially digested material also interacts with microbial activity, amplifying immune signaling. Microbial populations metabolize these substrates, producing compounds that can further influence the epithelial environment. These compounds may enhance or prolong immune responses, creating a feedback loop where incomplete digestion and microbial metabolism reinforce one another. The immune system responds to both the original substrate and the products of microbial processing, increasing the overall level of activation within the gut. This activation has functional consequences for digestion. Changes in barrier behavior can influence absorption, while alterations in motility can affect transit time and the distribution of substrates along the intestine. Fluid movement may be adjusted in response to immune signaling, modifying the consistency of luminal contents. These changes are not isolated effects. They are part of a coordinated response that attempts to manage the presence of material that has not been properly processed upstream. Immune activation in this context reflects a mismatch between substrate and system capacity. The intestine is encountering forms of material that is not designed to handle under normal conditions, and it responds accordingly. When upstream digestion is restored, the nature of the material reaching the intestinal surface changes. Smaller, fully reduced substrates interact with the epithelium through established transport pathways, reducing the need for immune engagement. The system returns to a state where immune activity is aligned with actual threats rather than with the byproducts of incomplete digestion. System-wide symptoms from upstream failure. When digestion begins below capacity, the effects are not confined to the gut. The digestive sequence supplies substrates, signals, and regulatory inputs to multiple systems, and when that sequence is disrupted early, the outputs that reach the rest of the body are altered in both quantity and form. Incomplete protein breakdown limits the availability of amino acids required for tissue maintenance, enzyme production, and signaling pathways. Impaired fat processing reduces the delivery of lipid components that support membrane structure and signaling. These changes shift the internal environment, and the body responds with adjustments that are experienced as symptoms across different systems. Energy regulation is one of the first areas affected. Nutrient absorption becomes inconsistent when substrates are not fully reduced, leading to fluctuations in the availability of usable fuel. The body attempts to stabilize this variability through hormonal adjustments that alter how energy is stored and released. These adjustments can produce patterns of fatigue, unstable energy levels, and changes in appetite. The underlying driver is not a lack of intake, but a mismatch between intake and the system's ability to convert that intake into accessible substrates. The immune system reflects these changes as well. Increased exposure to partially digested material and the byproducts of altered microbial activity raises baseline immune activation within the gut. Signals generated at the intestinal level extend beyond the local environment, influencing systemic immune behavior. This can manifest as a persistent state of low-level activation that affects how the body responds to additional challenges. The source of this activation is rooted in the material reaching the intestinal interface, rather than in an external input alone. Neurological signaling is also influenced by the state of digestion. The gut communicates with the central nervous system through neural and chemical pathways that respond to the condition of the digestive process. When digestion is inconsistent, these signals become less stable, affecting patterns of focus, mood, and perception of stress. The system interprets variability in nutrient delivery and immune signaling as changes in internal conditions, and neural output adjusts accordingly. This creates a link between digestive function and cognitive or emotional states. Fluid balance and circulation can shift as the body responds to altered digestive conditions. Changes in intestinal permeability, motility, and secretion influence how fluids are distributed and retained. These shifts can affect tissue hydration, circulation patterns, and the delivery of nutrients to peripheral systems. The body compensates for these changes, but the compensation reflects the underlying disruption rather than correcting it. Symptom clustering often emerges from this pattern of system-wide adjustment. Fatigue, digestive discomfort, variability in energy, and changes in immune or neurological function appear together because they share a common origin in upstream digestive failure. Each system is responding to the same set of altered inputs, and the overlap in symptoms reflects the interconnected nature of these responses. Restoring upstream function changes the inputs that drive these system-wide effects. When digestion proceeds with proper sequencing, nutrients are delivered in their intended forms, immune activation is reduced, and signaling between systems stabilizes. The body no longer needs to compensate for inconsistent inputs, and the pattern of symptoms begins to resolve as the underlying sequence is brought back into alignment.